All right, friends, we're here to take our second test. There we go. I'm going to blow it up a little bit. And so, like last time, I'm going to read each question to you. Then you're going to answer here in an ed puzzle. Okay, and you can always go back and redo a question. All right. So, question number one: enzymes that work in the stomach must adjust to the unique environment by being able to A, function in an acidic environment, B, function in a neutral environment, C, function in a basic environment, D, function in both an acidic and basic environment. Question 2. What occurs during the digestion of proteins? A, specific hormones break down proteins into simple sugars. B, specific enzymes break down proteins into amino acids. C, specific hormones break down proteins into complex starches. D, specific enzymes break down proteins into simple sugars. Question 3. The shape of a protein molecule is influenced by A, the sequence of amino acids in it, B, whether it is organic or inorganic, C, the number of chromosomes in a cell, D, the number of genes found in the nucleus. Question 4. The diagram below represents a process that occurs in organisms. A, broken down into B. Which row in the chart indicates what letters A and B in the boxes could represent? Right? So which one of these? Row A, starch is A, broken down into amino acids, B. B, protein, broken down into amino acids. C, starch, broken down into proteins. Or D, proteins broken down into simple sugars. Which one of those would be correct? Question 5. Which one of the following statements best describes enzymes? A, they slow down the rate of breathing. B, they are the building blocks of polymers. C, they influence the rate of chemical reactions. D, they speed up the conduction of impulses along a nerve cell. 6. Enzyme molecules normally interact with substrate molecules. Some medicines work by blocking the enzyme activity in pathogens. These medicines are effective because A, they are the same size as the substrate molecules. B, they have the shape that fits into all cell receptors. C, they have a shape that fits into the enzyme. D, they are the same size as the enzyme. Question 7. The diagram below illustrates a biochemical process that occurs in organisms. You've got sugar attached to a catalyst plus water makes simple sugars and the catalyst. The substance labeled catalyst is known also as A, an inorganic compound, B, an antibody, C, a hormone, D, an enzyme. Question 8. The diagram below represents the stages in the digestion of a starch. The products of digestion of starch shown would most likely contain A, minerals, B, amino acids, C, fats, D, simple sugars. Question 9. The graph below represents the effect of temperature on the relative rate of action for a particular human enzyme. So the x-axis is temperature, the y-axis is relative rate of enzyme action. What is the approximate optimum temperature for this human enzyme? A, 37 degrees, B, 41 degrees, C, 10 degrees, D, 60 degrees, C. Question 10. Which pH would indicate a substance that is more acidic than a substance with a pH of 4? A, 6, B, 2, C, 12, D, 4. The effective pH on a certain enzyme is shown in the graph below. The x-axis is pH. The y-axis is rate of enzyme action. At what pH would the enzyme be most effective? A, above 10. B, below 5, C, between 5 and 7, D, between 8 and 10. Question 12. The graph below represents the relationship between pH and the rate of enzyme action for two enzymes, X and Y. Which one of the following statements best describes this pattern of enzyme action? You can see we have pH on the x-axis, rate of enzyme action on the y-axis, and two curves. The curve representing enzyme X and the curve representing enzyme Y. A, at a pH of 7, the rate of action of enzyme Y is less than that of enzyme X. B, as the pH increases from 2.5 to 3.5, the rate of action of enzyme X decreases. C, as the pH increases from 10 to 11, the rate of action of enzyme Y increases. D, at a pH of 5.5, the rate of action of enzyme X is the same as that of enzyme Y. Question 13. In the graphs below, they show the relationship between the relative rates of activity of enzymes A and B and temperature. Graph 2 shows relative relationships between the relative rates of activity of enzymes A and B and pH. So the y-axis is rate of activity. The x-axis on graph 1 is temperature. The x-axis on graph 2 is pH. Under which conditions is enzyme A most effective? A, at 50 degrees C and a pH of 9. B, at 40 degrees C and a pH of 5. C, at 45 degrees C and a pH of 5. D, at 45 degrees C and a pH of 9. Question 14. The building blocks represented by letter A are, you can see in this chart, we've got 3 columns, lipids, carbohydrates, protein, and you've got 3 rows, building blocks, necessary elements, and functions. So we're looking for what would go into letter A here. A, glycerol and fatty acids. B, adenine and thymine. C, nucleotides. D, polysaccharides. Question 15. What type of molecule will be produced by the linking together of many molecules of glucose? A, hemoglobin. B, amino acid. C, starch. D, fatty acid. Question 16. The diagram below represents the synthesis of a portion of a complex molecule in an organism. Building blocks make a product. Which row in the chart could be used to identify the building blocks and product in this diagram? A would be row 1, B is row 2, C is row 3, D is row 4. Which best represents the building blocks and product from this diagram? Question 17. Mr. Kobach tested some rutabaga juice with iodine. It turned a dark black color. What does that tell you about rutabaga juice? A, rutabaga juice is an acid. B, rutabaga juice contains a lot of polysaccharides. C, rutabaga juice would make a great buffer. D, rutabaga juice denatures proteins. Question 18. Mr. Kobach added some salivary amylase to a starch solution. After waiting 20 minutes and letting it cook at 37 degrees C, he added Benedict's solution. Because the Benedict's solution changed from blue to orange, this tells Mr. Kobach A, he needs to buffer his saliva. B, his salivary amylase is not working correctly. C, the Benedict solution is a strong acid. Or D, the amylase must have broken down the starch into monosaccharides. Number 19. Why would somebody add enzymes to laundry detergent? A, to trick people into buying more of it. B, to reduce the pH of the laundry. C, to speed up the reactions that break down certain stains. D, to slow down the process of hydrolysis in your laundry. Question 20. Proteins are large biological molecules. Which of the following would not be considered a protein? A, pepsin, enzyme? B, insulin, a hormone. C, serotonin, a neurotransmitter. Or D, glucose molecules? 21. If a solution has a pH of 10, that means there are A, 10 times more H ions floating around it compared to a neutral substance. B, 10 times more OH ions floating around it when compared to something with a pH of 9. C, it has more monosaccharides than disaccharides. Or D, it must contain more saturated fatty acids than a standard carbohydrate. Question 22. On Halloween, Mr. Kobach got lots of candy. He went trick-or-treating while dressed up as Pepsin Man. When he tested the candy with iodine, the color did not change. What does this mean? A, he should not eat the candy. It's bad for his teeth. B, the candy only contains monosaccharides. C, the candy does not contain any polysaccharides. D, he should add a buffer to neutralize the candy. Question 23. Mr. Kobach placed a piece of liver in a test tube. When he added hydrogen peroxide, the solution began to fizz and bubble. What is the enzyme associated with the hydrolysis of hydrogen peroxide? A, amylase. B, pepsin. C, catalase. D, dehydrogenase. Question 24. In Mr. Kobach's enzyme comic, what was that fish with the helmet represent? The fish is saying, my job is to speed up the rate of reactions. I need to bind with a substrate. Oh, look, there's my friend Reactopus, the substrate. Sup, bud, are you ready? Hey, check out my active site. Nice. A, the fish represents A, an enzyme. B, the substrate. C, the induced fit. D, it's impossible to tell. Question 25. What happens when a hydrolytic digestive enzyme catalyzes a reaction? A, reactants are placed together while water is removed. B, nothing actually binds to the active site. C, the substrate becomes denatured. D, the reactant substrate is split into smaller pieces. Question 26. Scientists worry about the acidification of ocean water because A, it's harder to drink acidic ocean water. B, more acidity in the ocean will lead to more hurricane activity. C, the reduction of pH affects enzymes that allow coral reefs to form. D, the acidity will burn a hole in even the thickest wetsuit. Question 27. People who are considered lactose intolerant do not digest milk sugars because A, they are also vegetarians. B, they do not make any lactase enzyme in their intestines. C, they make too much pepsin in their stomachs. D, they do hydrolysis of lactose when they should be doing dehydration synthesis. Question 28. Which characteristic allows enzymes to function in a specific way? A, each enzyme has a characteristic shape. B, enzymes are long, complex fats. C, enzymes are complex compounds composed of starch. D, each enzyme is made up of 4 subunits. Question 29. Hydrogen peroxide, H2O2, is a toxic compound that is produced by plant and animal cells. These cells also produce the enzyme catalase, which converts H2O2 into water and oxygen gas, preventing the buildup of H2O2. The student designed an experiment to test the effect of an acidic pH on the rate of the reaction of H2O2 with catalase. The data below summarizes the outcome of the experiment. You can see that there's a data table here with 4 different pH levels, 7, 6, 5, and 3, and then 4 different reaction rates. 1.5 milliliters of oxygen per minute, 1.3, 1.0, 0.55. The best explanation for the change in catalase activity as the pH changed from 7 to 3 in a given experiment is that A. Decreased oxygen production causes catalase to increase the rate of reaction. B. The student most likely cooled the O2 solution, causing the reaction rate to increase. C. Strong acids digest the catalase, causing the reaction to increase. D. In acidic solutions, the shape of the catalase changes, causing the reaction rate to decrease. Question 30. In the diagram below, X represents a process that causes protein to unfold and stop functioning. Process X is most likely caused by A. The digestion of amino acids that make up the proteins. B. The synthesis of a protein with different simple sugars. C. Removal of the gene that codes for the production of that protein. D. An internal factor in a body, such as temperature, has increased. Question 31. Which of the following would be considered a polymer? A. A starch molecule. B. A glucose molecule. C. An amino acid molecule. Or D. A glycerol molecule. Question 32. Which one of the following statements is most likely correct regarding soil bacteria that inhabit the polar regions of the Earth? A. They do not carry out processes of respiration. B. They contain enzymes that function at very low temperatures. C. They are part of the abiotic resources of that ecosystem. And D. They obviously do not reproduce. And our last question on the test. One effect of uncontrolled diabetes is that the blood might develop an acidic pH. As a result, cells may not be able to regulate their internal pH. Within these cells, this could cause a disruption of the function of the biological catalysts known as A. Enzymes, B. Antibodies, C. Toxins, or D. Antigens. And that completes the test. Feel free to go back and work on any other questions before you turn this in. And thank you. Good luck and Godspeed.